Ini berapa? Itu 13. Ganti. Ini di luar. Di luar kok. Ya, Bu. Terima kasih.
Be, bangun Be ke sana, Be.
Iya.
Cepet.
Ambil cusan sampai.
Good morning and warm greeting to everyone. First of all, Honorable Prof. Amalka and Dr. Retnat Vivi Fitriah Masduki as the lecturers of the Capital Selecta course. Respected Prof. Kohsuke Sensei as our reputable speaker for today, and Honorable Devianti Nur Afifa as our moderator, and all participants present in the classroom as well as those joining us via Zoom. We are grateful for the opportunity to gather here today in good health to attend the lecture series on biochemistry and biomolecular engineering with the topic in vitro reconstitution of synthetic metabolic pathway using thermophilic enzyme. Before we begin, allow me, Salwa Fania Putri, to guide this class as your master of ceremony. Here's the agenda for today's session. First, opening remarks, and then a lecture presentation by our esteemed speaker, Prof. Kohsuke Honda, a professor at the International Center for Biotechnology in Osaka University. And the third, Q&A session, which will be moderated by Devianti Nur Afifa, and the last is closing. We hope this session will bring valuable insight and enrich our knowledge. Without further ado, let's begin the event. Now hand over the session to our moderator, Devianti Nur Afifa, to get the next agenda.
Okay, thank you, Salwa. Once again, I would like to thank you and welcome back to Professor Honda to this guest lecture event. And also thank you for all participants for the guest lecture series on biochemistry and biomolecular engineering. Please allow me introduce myself. My name is Devianti Nur Afifa, the moderator for this session. Today we have an exciting and relevant topic, in vitro reconstitution of synthetic metabolic pathway using thermophilic enzymes. Before we begin Professor Honda's presentation, for those who have just joined, I will briefly read Professor Honda's CV again. Professor Kohsuke Honda, he received his PhD in 2003 from Kyoto University, Japan, under the supervision of Professor Sakayu Shimizu. Following his doctoral studies, he continued as a postdoctoral fellow in the Shimizu Lab. In 2005, he joined the Department of Biotechnology at the Graduate School of Engineering, Osaka University, as an assistant professor. He was promoted to associate professor in 2010 and advanced to full professor at the International Center for Biotechnology, Osaka University in 2019. Currently, he also serves as an adjunct professor at the Faculty of Mathematics and Natural Science, Institut Teknologi Bandung, Indonesia, and the Faculty of Science Chulalongkorn University, Thailand. Professor Honda's research focus on applied microbiology and synthetic biology with a particular interest in the application of extremophils and their enzyme. His contribution to the field have been recognized with several awards, including the Amino Enzyme Application Award in 2013 and Young Scientist Award from the Japanese Society of Enzyme Engineering 2015, the Japan Bioindustry Association in 2017, and the Society for Biotechnology in 2018. He has published more than 100 original papers, including 13 co-authors with Thai researchers. Currently, Professor Honda serves as a member of the steering committee for IC Biotech and holds the position of chair at the International Center of Biotechnology. Okay, all right, Professor Honda, are you ready for the presentation?
Yes, thank you very much.
So please, this time is yours.
Okay, thank you very much. And thank you very much for the good introduction. And yeah, actually, I'm sorry that in my introduction, I didn't mention about, I mentioned only about my, the co-author on paper with Thai researcher, but actually I also have some the co-authored paper with the Indonesia, particularly ITB, including Professor Amaroka. But anyway, so you may remember in the last week I talked about somewhat the general topics, how we can apply our the research output to the practical application in the commercial scale. And in this week, in this lecture, I would like focus more on my own research topics rather than the general the topics on biotechnology. So now let me share my screen just a moment. Now, can you see the slide?
Yes, yes.
Is this okay?
Maybe you can make in a slideshow so it will be bigger.
Just a moment. No. Okay.
Yes, it's good.
Yes. Okay, so now let me start my lecture. So as introduced by the moderator today, I will talk about the in vitro reconstitution of metabolic pathway using isolated enzymes, which means by assembling isolated or purified enzymes. Now we are trying to make a kind of the enzyme cascade, which is mimicking the natural or artificial metabolic pathway, and to apply those reconstituted metabolic pathway to the chemical synthesis. So this slide is the one I used in my last lecture. So in the last lecture, I introduced the application of this enzyme, the D-pantlactone hydrolase or lactonase, to enantioselective production of this compound, D-pantoic acid, which can be used as a precursor of coenzyme A or related compounds. And before the discovery of this enzyme, this compound was synthesized by organic chemistry based method. That after the discovery and the development of the enzymatic process using this DPL hydrolase, finally they achieved the marked reduction of the consumption of the water, the salt, or organic solvent, and more importantly, they succeeded in the reduction of energy consumption in terms of the carbon dioxide emission. So in this way, application of biotechnology often give us not only the economic, but also ecological benefit for our society. And this is an example of the application of isolated enzyme. But you may know nowadays, we can also produce a variety of useful compound by using the fermentation technology with the living organisms. So actually, some of you, particularly in the Muslim countries, may have a bit negative impression about the fermentation because fermentation technology is also used for the alcohol fermentation. But actually, not only alcohol beverage, the fermentation technology is used for the production of the many useful compounds. For example, maybe you know this one, MSG, monosodium glutamate, or Ajinomoto, which is a famous flavor enhancer produced by a Japanese company, Ajinomoto. So this compound, this is actually amino acid. This amino acid, glutamic acid, is also produced by fermentation using the Corynebacterium glutamicum. And nowadays, some amino acid producing company, including Ajinomoto, are using metabolically engineered C-glutamical for more efficient production of Ajinomoto, the flavor enhancer. And this is another Japanese company, Kaneka. This company is engaged in the production of biodegradable plastic, which is produced by special microorganisms. And the actual chemical name of this bioplastic is something like this: 3-hydroxybutanoate and 3-hydroxyhexanate. But anyway, so this kind of fermentation-based plastic has been more widely used as a practical alternative to the petroleum-based plastic. And these are the product of Japanese fermentation company, not only Japanese fermentation or chemical company. But if we look at other country like US or European country or a Chinese company, they also produce many useful compound or materials by using the fermentation technology. Now, for example, the DuPont, this is a very big chemical company, produced with this compound 1,3-propanediol, which can be used as a, the precursor of this plastic resin, so-called Sorona, the biopolymer, by using metabolically engineered E. coli. And this is a product of the amylase company, the artemisinin. So this is, the kind of the drug, anti-malaria drug. And originally, this compound was found from Chinese medical herb, Artemisia annua. But of course, this is a plant, and it takes very long time to be cultivated. So what the amylase person did, amylase researchers did, is so they isolated the genes involved in the biosynthesis of this compound in this medical herb, and they transferred those genes into microorganisms, in their case, the baker's yeast. Then the engineered baker's yeast started to produce this anti-malaria drug instead of the medical plant. And of course, the cultivation time for the yeast is much, much shorter than that for the medical plant. So they succeeded in the production of this artemisinin compound more quickly and more abundantly. So in this way, metabolic engineering has already widely employed for the practical production of this kind of useful compound. But actually, we also have to consider about some technical limitation of the metabolic engineering. For example, in this slide, now you can see the outline of the metabolic pathway in the typical heterotrophic microorganisms. And if you want to produce ethanol from glucose more efficiently by the metabolic engineering of these microorganisms, so what kind of engineering you can introduce in this pathway? So maybe the most straightforward way is to stop these reactions, knock out or knock down these reactions, this metabolic reaction. Then the glucose taken up the microorganisms will be converted into ethanol without being rooted into undesired metabolic pathway or unexpected metabolite. But maybe you can notice that, for example, in this pathway, pentose phosphate pathway, they produce some precursors for the biosynthesis of nucleic acid, like DNA or RNA. And as you know, a TCA cycle is involved not only in the energy production, but also the biosynthesis of the amino acid or related compound. And acetyl-CoA is a very important, the primary precursor for the lipid biosynthesis. And the nucleic acid, amino acid, or lipid, all of them are indispensable metabolite for living cell. So if we stop, we knock down or knock out this. It's okay. So if we knock down this metabolic reaction, then the host microorganisms cannot produce this metabolite, which means that microorganisms, the microbial cell, cannot be alive anymore. So we cannot introduce such a metabolic engineering into a living cell. So in other words, I think the metabolic engineering is a kind of tug of war between microorganisms and human beings. So they are microorganisms and they are human beings. So what is important for microorganisms is to grow or to survive. On the other hand, what we human beings are interested in is the production of the commercially valuable chemical. So what they want to produce, what microorganisms want to produce is for their growth or for their survival, and what the human beings expected microorganisms to produce is the commercially valuable product. So, the desired compound of microorganisms and human beings are not always the same. So it is very important to find the good compromise or a good balance between the, what the microorganism want to produce and what human beings want to produce. Or another approach may be to cut this tug. I mean, to separate the metabolic pathway for chemical synthesis from the physiological activity of microorganisms. The physiological activity means such as growth or survival of microorganisms. So how we can separate this? So what we can do is to construct or reconstitute the metabolic pathway outside of the cell. For example, we cannot knock down this metabolic pathway in living cell, as I said. So instead of knocking down this metabolic reaction, an alternative approach may be to isolate these enzymes from the living cell and mix them together in a bioreactor, then put the glucose. So the glucose put in the bioreactor will be converted into ethanol with 100% conversion yield. So this is what we can do to separate the metabolic pathway for chemical synthesis from physiological activity of microorganisms. But you may notice that isolate only the enzyme of our interest is sometimes very time consuming. For example, even in a very simple microorganisms like E. coli, they possess the thousands of enzymes inside of their cells. And among those thousands of enzymes, we have to isolate only our desired enzyme. So this procedure is, of course, very time consuming. So to skip this time-consuming procedure for enzyme purification and isolation, we focus on the enzymes from thermophilic microorganisms or thermophiles. The thermophiles are the group of microorganisms which grow at very high temperature. So similarly to Indonesia, Japan is also located in the volcano area. We have many volcanoes and also the hot spring. And if we go to the hot spring and take some sample and cultivated them at high temperature, we can often obtain this kind of thermophilic microorganisms. So actually, those of these microorganisms, all of these microorganisms have been isolated in Japan. So for example, this one, the thermophilus, so this is the thermophilic bacterium. So this bacterium has an optimum growth temperature of around 75 degrees Celsius, which is isolated in the Mine hot spring area in Shizuoka Prefecture of Japan. And maybe some of you may know this thermophile. So this Thermococcus codacalensis, and this is not bacteria. This is this micro thermophile belongs to archaea. So this archaea has an optimum growth temperature of 85 degrees, and which was isolated in the Kodakara Island in Kagoshima Prefecture of Japan. And the DNA polymerase obtained from this thermophilic archaea is now widely used as the polymerase for PCR, polymerase chain reaction. So maybe some of you know, some of you have used DNA polymerase provided by a Japanese company, Toyobo. The product name is KOD or KOD1. So the KOD1 DNA polymerase is actually originally obtained from this thermophilic archaea. And this is also archaea, belongs to archaea sulfurous tocodi, which has optimum growth temperature of 80 degrees C. And what is interesting for this thermophilic archaea is, so this is not only the thermophile, but also to be acidophile. So not only the very extreme optimum growth temperature, this thermophilic and acidophilic archaea has an optimum growth pH of 1 or 2. So it can grow in very high temperature and low high acidity, low pH condition. And this one is also isolated in a Japanese hot spring in Hakone area. So because these microorganisms are adapted to a high temperature environment, the enzymes produced by these microorganisms also adapted to high temperature. Usually, the most of the protein produced by the mesophilic means the microorganisms growing at around room temperature, like E. coli, Baker's yeast, Bacillus, Pseudomonas. This kind of typical microorganisms are not adapted to high temperature environment. So the enzymes produced by those normal mesophilic microorganisms are denatured if they are incubated at high temperature, like 70, 80, or 85 degrees Celsius. But the enzymes and the proteins produced by these thermophilic microorganisms can tolerate to such high temperature environments. And another important point is such a very excellent thermal stability of thermophilic enzymes are attributed to their amino acid sequence. I mean, there is no special the mechanisms inside of these thermophilic cells to make their enzyme to be thermotolerant. The thermotolerance, the excellent thermotolerance of the thermophilic enzymes simply comes from the amino acid. So which means as long as the amino acid sequence is identical, regardless of the host strain used for enzyme production, the resulting recombinant enzymes are still thermostable. So if we isolate the genes DNA encoding the thermophilic enzyme from thermotolerant microorganisms and introduce those genes in mesophilic host, for example, E. coli, because DNA is identical, the amino acid sequence of the recombinantly produced enzyme are also identical with those from the original host thermophilic microorganisms. So which means even if they are produced in the recombinant mesophilic host like E. coli, the recombinant thermophilic enzymes are still thermostable. On the other hand, the enzymes, proteins, which originated from E. coli is not stable at a high temperature. So just by incubating the lysate, crude lysate of E. coli at a high temperature, like 70 or 80 degrees Celsius, the E. coli enzymes are totally denatured. On the other hand, the thermophilic enzymes are still active. So without time-consuming procedure like the ammonium sulfate precipitation or column chromatographies, we can very quickly and simply purify the thermophilic enzyme just by the heat treatment of the crude lysate of recombinant E. coli. And finally, by assembling these recombinantly produced thermophilic enzymes, we can make an enzyme cascade, which is mimicking the natural metabolic pathway or artificial metabolic pathway for chemical synthesis. So this is our basic strategy to produce, reconstitute metabolic pathway outside of the cell in vitro by using thermophilic enzymes. Now, let me introduce some example of our in vitro metabolic pathway, the metabolic pathway reconstituted outside of the cell. So you can see this is a glycolytic pathway or Enden-Meyer-Hof partners pathway, which you can find in every biochemistry textbook. So you may wonder if we can reconstitute this metabolic pathway by using thermophilic enzyme. The answer is definitely yes. Yeah, we can purify these enzymes by the heat treatment of the crude lysate of E. coli like this, and by assembling them together, we can reconstitute this glycolytic pathway outside of the cell. But if we want to apply this in vitro reconstituted pathway for the production of pyruvic acid from glucose, what would happen? Now, of course, the glucose is a very cheap compound, and pyruvate is somewhat more expensive. So producing pyruvate from glucose may be economically feasible. But to do that, not only glucose, we also have to put some cofactor like ADP or NAD. And these compounds are very expensive. So total production cost through this reconstituted pathway is not very feasible. To produce 2 molecules of pyruvic acid, we have to discard 2 molecules of ADP or 2 molecules of NAD. So how we can overcome this problem of cofactors? So to overcome this challenge, what we focus on is this kind of, the kind of unusual, the modified glycolytic pathway, which are found in some thermophilic archaea. So, this green arrow shows the very unique enzyme found in this archaea modified glycolytic pathway. So, for example, the initial reaction of glycolysis, the phosphorylation of glucose to glucose 6-phosphate catalyzed by hexokinase. The usual enzyme use ATP as a phosphate donor for the phosphorylation of glucose and release ADP as a as a reaction product. But you can see in this archaea modified pathway, their enzyme use ADP but not ATP and release AMP but not ADP. And another unique point is here, in the bacteria or eukaryotic glycolytic pathway, the oxidation of GAP, glyceraldehyde 3-phosphate, into 3-phosphoglycerate is catalyzed by these 2 enzymes, 2 distinct enzymes. The one is catalyzing the GAP oxidation of GAP into 1,3-BPG by using NAD, and in the second reaction, this compound is converted into 3-PG by phosphorylating ADP into ATP. So actually, this is the ATP producing reaction, the very important reaction in glycolysis. But in archaea pathway, they skip this ATP producing reaction. Only one enzyme is required for the oxidation of GAP into 3-PG. Instead, in this archaea modified glycolytic pathway, they produce ATP from AMP, but not ADP, directly from PEP to yield the pyruvate. But the point is, although they use this kind of very unique enzyme with unusual cofactor specificity, the total stoichiometry of this archaea modified pathway is essentially same with the normal classic glycolytic pathway found in bacteria or eukaryotes. So they also use 2 molecules of ADP and generate 2 molecules of ATP. And they use 2 molecules of NAD, 2 molecules of NAD, and 2 molecules of NADH or NADPH, like this. So even if we employ this archaeal modified glycolytic pathway, we still have this problem of cofactor imbalance. But what we did is, because we can reconstitute metabolic pathway outside of the cell, so we can flexibly choose what kind of enzyme can be assembled, reconstituted to construct an artificial metabolic pathway. So we can freely design the metabolic pathway. So actually what we did is, we exchanged these two reactions, exchanged these two enzymes involved in this two-step reaction with this green one, like this. So now ATP is consumed in this reaction and in this reaction. So in total, 2 molecules of ATP are required. But at the last step of the reconstituted pathway, 2 molecules of ATP are obtained from 2 molecules of ATP. So, the consumption and the production of ATP and ADP are totally balanced. So only if we put very small concentration of ATP to initiate the reaction, then those ATP or ADP are continuously recycled in the pathway. So if we look at the overall stoichiometry, there is no production or consumption of ATP. So only with a very small amount of ADP or ATP that we can implement this metabolic pathway. And actually, we also integrate one more enzyme, the kind of lactate dehydrogenase, which can catalyze the reduction of pyruvate into lactate using NADH. So not only the balance between ATP and ADP, the balance between NAD and NADH could also be achieved. So just by putting the small concentration of this cofactor, this pathway was continuously carried out. So by using this in vitro reconstituted pathway, we could achieve the production of lactate from glucose with an overall conversion yield of 100%. And as we expected, ATP could be continuously recycled, and the estimated recycling number was more than 30. So this is one of the example of our reconstituted pathway. And after the development of this non-ATP forming glycolysis, we also tried to extend this artificial glycolytic pathway to the production, for the production of different compounds. In this case, we tried to produce 1-butanol, which is expected to be our next generation biofuel. So to achieve this, we assembled 16 enzymes in a single reaction mixture. And actually, this butanol production pathway was also designed based on the natural metabolic pathway involved in the butanol production. But actually, we introduced some artificial modification in this pathway. that is here from crotonyl-CoA to butyraldehyde. So in the natural butanol producing microorganisms, Clostridium acetobutylicum, this intermediate, crotonyl-CoA is converted to butyraldehyde by using these two enzymes, butyryl-CoA reductase and aldehyde dehydrogenase. So this black arrow shows the natural metabolic pathway in Clostridium. But we found this kind of reaction hardly proceed in vitro. The one problem is this enzyme was very sensitive to oxygen. So in the presence of oxygen, this enzyme is rapidly inactivated. And another problem is this enzyme requires this partner protein, so-called redox partner. So actually, this is a reducing reaction. So that this enzyme catalyzes the reduction of this CC double bond. And because this is a reducing reaction, of course, they need some reducing power. And in case of this enzyme, they use NADH as a reducing source to catalyze the saturation of this CC double bond. But interestingly, this enzyme cannot accept reducing power electron directly from NADH. Instead, they utilize this partner protein to transfer the electron from NADH to this enzyme. So which means we need this partner protein, which are also sensitive to oxygen stress. So it is very challenging to operate this kind of in vitro reaction under anaerobic, no oxygen environment. So we gave up to use this enzyme. Instead, what we focused on was this enzyme, aldehyde dehydrogenase. So this enzyme catalyzed the reduction of this compound butyryl-CoA into butyraldehyde, accompanied with the release of CoA, coenzyme A moiety. And actually, it was well known that this kind of enzyme, aldehyde dehydrogenase, substrate specificity of this type of enzyme is not very strict. Actually, butyryl-CoA and crotonyl-CoA have a very similar structure to each other. The difference is only this CC double bond. Crotonyl-CoA has the one CC double bond here, but butyryl-CoA, this double bond is saturated into a single bond. So we expected that maybe this aldehyde dehydrogenase can accept not only butyryl-CoA, but also this compound, crotonyl-CoA. Actually, we investigated the substrate specificity of this aldehyde dehydrogenase obtained from thermophilic bacterium Thermophilus. So as you can see, of course, this enzyme shows higher activity to this butyryl-CoA, but this enzyme also shows some acceptable level of the activity against this compound, crotonyl-CoA. So we decided to use this aldehyde dehydrogenase to convert crotonyl-CoA into this intermediate croton aldehyde. So then what we have to do next is saturation of this CC double bond to make butyraldehyde. And to achieve this, we focus on totally different enzyme, the in reductase, which can catalyze the saturation of CC double bond, which is conjugated with the carbonyl residues, like carboxyl residues or keto residues or aldehyde residues. So you can see these CC double bonds are conjugated with the carbonyl group. And actually, we found some homologue of in reductase in some thermophilus genome. And when we checked the activity of this enzyme against this croton aldehyde, you can see that this C double bond is also conjugated with the aldehyde group. So this compound can be a substrate for this enzyme. And yeah, luckily we found that this enzyme can catalyze the saturation of this CC double bond. So finally, we designed this kind of artificial metabolic route to convert protonyl CoA into butyraldehyde. So actually what we did is we just changed the order of the reaction. So from protonyl CoA to butyraldehyde, they require two steps of reducing reaction. The first one is a reduction of CC double bond, and the second one is a reduction of this carboxyl group into aldehyde group. So in our designed pathway, we changed this order. We first reduced the carboxyl group into aldehyde group. Then we reduced this CC double bond by using this in reductase. So finally, okay, and finally, we designed this pathway and we reconstituted this artificial pathway by assembling the 16 the thermophilic enzymes together. And this is the result. And actually, the conversion yield was unfortunately not 100%. So this was probably due to the, some thermal degradation of some intermediate or something. But anyways, we could obtain some amount of butanol from glucose. And another good interesting point of our in vitro metabolic pathway is because our pathway is constructed by the isolated enzyme. So by measuring some kinetic parameter like Km or Kcat of each enzyme, so we can simulate the actual metabolic flux through our designed pathway. So we predicted the time profile of intermediate in our in vitro metabolic pathway using the computer model, using the kinetic parameter of the enzyme. And actually, so this model prediction was in good agreement with the actual the intermediate time profiles, which was measured by the actual monitoring of reaction mixture. So which means our the in vitro reconstituted pathway can also be available for the fine tuning of metabolic flux. But anyway, in this way, we have constructed a number of in vitro metabolic pathways using the thermophilic enzyme so far, and we succeeded in the production of this compound from glucose or glycerol or even from chitin. But not only the design and construction of this kind of in vitro metabolic pathway, we are also interested in development of some supporting technology to increase the feasibility, practical availability of our in vitro metabolic pathway, like enzyme immobilization or directed evolution or cofactor regeneration and stabilization. So in this lecture, I like to some example of this kind of supporting technology in the remaining time. So first, I like to talk about these topics, directed evolution. So actually, in my last lecture, I already talked about the basic concept of directed evolution, which was originally developed by the Francis Arnold, who received the Nobel Prize in Chemistry in 2018. So by using, for example, the error-prone PCR or this kind of the mutagenesis technique, we introduce the random mutation in the DNA, the genes encoding an enzyme. And by overproducing the translated product of these mutation genes, we can obtain a library of the mutant enzyme in which they have some amino acid substitution in different position. And actually, most of them shows lower activity compared with the wild type enzyme. But if sometimes some of them exhibit the better activity or better stability than the wild type enzyme. So through the some high throughput assay, we can select the enzyme which has improved properties compared with the wild type. Then after choosing this kind of potential mutant, we can subject these promising mutant enzyme into another round of mutagenesis and selection. So by repeating this, we can improve the performance of the enzyme. In this case, we tried to, they tried to improve the thermal stability of the enzyme. And finally, we could achieve the improvement of the half-life time in this way. So this kind of directed evolution can be used, sometimes used for the improvement of the enzyme property. So in your case, because we are using thermophilic enzyme, we still have some difficulties to find the good enzyme from thermophilic sources. The one example was this enzyme, pyruvate decarboxylase, which convert the pyruvate into acetaldehyde. So this enzyme play a very important role to connect. The pyruvate is actually the final product of glycolytic pathway. So to connect glycolytic pathway to another metabolic pathway for the biosynthesis of some useful compound, this enzyme plays a very important role. But even if we carefully we carefully looked for the pyruvate decarboxylase from thermophilic source using BRAS or other database searches. But finally, we could not find any hit in the BRAS search. I mean, there is no pyruvate decarboxylase in thermophilic microorganisms. Instead, when we subject the pyruvate decarboxylase to BRAST homology search, we have many homologous protein, but all of them was annotated to be this kind of different enzyme, acetolactate synthase. You know, we looked for this enzyme, pyruvate decarboxylase, but BLAST database suggests us this different enzyme, acetolactate synthase. So what kind of chemical reaction this acetolactate synthase catalyze? So they catalyze such a reaction. They use 2 molecules of pyruvate as substrate and produce acetolactate with an emission of 1 molecule of carbon dioxide. So you can notice that this reaction catalyzed by pyruvate decarboxylase and this enzyme catalyzed by acetolactate synthase is somewhat similar. The difference is the number of substrate molecules and the product. So yeah, actually, if not only amino acid sequence, but we look at the 3D structure of the proteins, both pyruvate decarboxylase (PDC) and acetolactate synthase (ALS) shows the very similar structure. And actually, some literature said the reaction mechanisms is also very similar to each other. So first, they accept first pyruvate molecule, which is introduced in the catalytic center and make some conjugation with this cofactor TPP to produce this kind of intermediate. Then carbon dioxide is released, and they make this kind of transient intermediate. And afterwards, if the proton attacked this intermediate, then they produce acetaldehyde. But if another molecule of pyruvate comes to this transient intermediate, then the product will be acetolactate. So only the difference between the pyruvate PDC and ALS is only the difference in the second substrate which attack to this intermediate. So although we could not find any pyruvate decarboxylase from our our library of thermophilic microorganisms, we could found many acetolactate synthase (ALS) from some files. So what we tried to is not the improvement of the thermal stability of this enzyme, but we tried to change the substrate specificity of thermophilic ALS to make like that of PDC, pyruvate decarboxylase. Maybe I can show some result. So this is the activity of wild type acetolactate synthase. So if we put pyruvate in, if we incubate this wild type ALS and pyruvate, of course, they produce acetolactate. But after some directed evolution experiment, we obtained some mutant enzyme which has improved acetaldehyde forming activity and decreased activity of acetolactate formation. So actually, the reaction specificity of this mutant enzyme is still not perfect. But anyway, we could produce an enzyme which can catalyze the acetaldehyde formation at a high temperature environment. So actually, this is a result of thermal stability. So, you know, both wild type ALS and our mutant ALS show the very nice thermal stability they retained almost perfect activity even after the incubation to 60 degrees for 19 minutes. On the other hand, this is PDC, pyruvate decarboxylase, obtained from mesophilic microorganisms, quickly lost their activity at 60 degrees. So in this way, not by improving the thermal stability, but by changing the thermal stability of, not by changing the substrate specificity of, neighbor-related enzyme, we succeeded in to produce the thermophilic enzymes with desired catalytic ability. Okay, so this was the story of our directed evolution of a study. So next, I like to introduce this example, the reaction in organic solvent. So just a moment. If you remember, this is the basic strategy of our in vitro metabolic pathway reconstitution. So by introducing thermophilic enzymes into mesophilic host, we can quickly obtain the heat purified enzyme by the the incubation of recombinant cell at high temperature. And usually we use E. coli. But as long as these host microorganisms does not have thermophilicity, thermal stability, any kind of microorganisms other than E. coli can also be used. So my next story is the use of different microorganisms, different bacteria as a host of the recombinant thermophilic enzyme production. So in this study, we use this bacteria, Rhodococcus opacus, instead of E. coli. Now you can see, this is actually a movie. So in the left flask, we put the wet cells of Rhodococcus opacus, and in the right flask, we put E. coli cells. And we suspended those wet cells of Rhodococcus and E. coli by magnetic stirrer in organic solvent, in this case, cyclohexene. So they are not water, they are organic solvent. So as you can see, E. coli or most of microorganisms cannot be suspended in organic solvent because their cell surface is hydrophilic. But interestingly, the Rhodococcus opacus cells can be suspended even into organic solvent. So if we look at the suspension of Rhodococcus opacus cell in organic solvent, we can see they are suspended in a form of the small aggregate. I mean, they are not suspended unicellularly, not in the form of single cells, but they are making some small aggregation. And these small aggregations are suspended in the organic solvent. So you can see this is a scale bar. So this small aggregate has around 100 meter diameter, but actually the diameter of bacterial cells is typically 1 micrometer or 2 micrometer. So you can see they are not single cell, they are the aggregation of cells. But anyway, because they are suspended as a small aggregate in organic solvent, which means they have a larger interfacial area between microbial cell and organic solvent. So if we put those Rhodococcus opacus cells in organic solvent containing water insoluble, but organic solvent soluble substrate, because they have a larger surface area. Maybe you can understand from this video. So because they are suspended in form of small aggregation, there are many small aggregation, which means they have a larger surface area between the solvent and cell. So this kind of increased interfacial area is very important to increase the driving force for the water insoluble compound to penetrate it, diffuse into the cells. So actually inside of the cells are, the kind of the aqueous water environment. So if we put some enzyme, if we express some enzyme inside of Rhodococcus opacus cells, they are still active. And they also have some NADA or NADH. So by putting these wet cells of Rhodococcus opacus overproducing this thermophilic enzyme in our organic solvent containing this substrate, then we could facilitate the reaction conversion of this kind of water insoluble compound. And actually, so this is the reaction condition. We put 1700 milligram of the wet cells into 700 microliter of organic solvent, cyclohexanol, containing 1 molar of this substrate, ketoisoforone. And this is our target reaction, the enantioselective reduction of ketoisoforone into 6R ribosome. So actually, this can also be used as a precursor for the pharmaceutical biosynthesis, pharmaceutical chemical synthesis. And you can see here, when we use whole cell catalyst suspended in the organic solvent like cyclohexanol, we could achieve more than 60% conversion of ketoisoforone into 6R ribosome. You can see this yellow triangle shows enantioselectivity of the product. So the enantiomer excess value of the product is about 90%, pretty high. And compared with the conventional, this kind of the two-phase bioconversion system, the conversion yield is not as high as the case of the whole cell reaction, because the interfacial area between the enzyme solution and the substrate solution is limited. So in this way, by employing some microorganisms with a unique property, which is not seen in E. coli, we are also able to expand the applicability of the thermophilic enzyme, like in this way. Okay, so for next, I'd like to talk about these topics, ATP regeneration. Actually, this study was performed by my international student who comes from Indonesia. So as I talked in the introduction of our non-ATP forming glycolytic pathway, the cofactor balance is very critical to implement the in vitro metabolic pathway. And in case of glycolytic pathway, we could achieve the cofactor balance by designing artificial glycolytic pathway. But sometimes it is difficult to design such a cofactor balance pathway. In such case, we may have to put some ATP degeneration reaction or NADH degeneration reaction. Actually, so ATP is widely involved in a variety of commercially important enzymatic reaction. So until now, the ATP degeneration means the ATP production from ATP, the phosphorylation of ATP. The many ATP regenerating enzyme has been proposed. For example, this reaction, this enzyme creatine kinase catalyzes the regeneration production of ATP from ADP by using this substrate, phosphocreatine. And this one, acetate kinase, similarly, and this acetate kinase use this substrate, acetyl phosphate, for the phosphorylation of ADP to produce ATP. And actually, so this enzyme has a very nice activity. But the problem is, as you can see here, the price is very expensive. We are sorry that the price is shown only in Japanese yen. But maybe you can imagine, so both of them are very expensive compound. Instead, this compound, polyphosphate, this is a polymer of inorganic phosphate, is considerably cheaper than this compound. So you may think that we can use this enzyme, this substrate and this enzyme polyphosphate kinase for the ATP reduction. But the problem of this enzyme is okay, but the problem of this substrate is, polyphosphate is the polymer of the phosphate. And you may know the phosphate makes some insoluble salt with some kind of divalent metal ion, like magnesium or manganese. And actually, some enzymes requires the magnesium or manganese. Actually, some enzyme, including kinase itself, requires magnesium as a cofactor to exert their enzyme. I mean, ATP and magnesium make some complex, and this complex is accepted by the kinase as a substrate. So the magnesium ion is indispensable for this enzyme. Nevertheless, if we put high concentration of polyphosphate in the reaction mixture, the polyphosphate and magnesium ion make an insoluble salt, which cannot be used enzyme. So this is a very nice enzyme reaction using considerably cheap phosphate toner. Nevertheless, it is still challenging to employ this enzyme in the practical operation of in vitro enzyme reaction. So what we did for the ATP regeneration in our study is we constructed this kind of artificial glycolytic pathway, so-called non-oxidative glycolytic pathway. Actually, this pathway was originally reported by US group more than 10 years ago in Nature paper. And the point is they use starch as the starting material. And the starch is decomposed into acetate. So 1 molecule of glucose, 1 glucose moiety is converted into 3 molecules of acetate. And at the last step of this reaction, there is the ATP producing enzyme. Actually, this is identical to this one, acetate kinase. Anyway, so through this metabolic pathway, they can supply ACP, acetyl phosphate, an expensive substrate for this kinase. So we can use this metabolic pathway for the production of ATP from ADP. And the unique point of this pathway is, actually, they use starch. Actually, starch is a polymer of glucose, as you know. So they use glucose as a substrate and produce acetate. But from 1 molecule of glucose, they can produce 3 molecules of acetate. So if we consider the usual glycolysis is always accompanied with carbon dioxide emission, catalyzed by, for example, pyruvate decarboxylase, which I mentioned, which I introduced in the directed evolution study. Usually in living cell, decompose glucose C6 compound into C2 compound like acetaldehyde, ethanol, or acetyl-CoA. But in this pathway, they produce 3 molecules of C2 compound without emission of carbon dioxide. So which means there is no oxidative. Oxidative means CO2 emitting reaction, non-oxidative glycolysis. So the mechanisms why this metabolic pathway can catalyze non-oxidative glycosylation is in this carbon rearrangement module. So in this carbon rearrangement module, the 3 molecules of C4 compound, erythritol 4 phosphate, is used as a substrate. And after the series of carbon rearrangement reaction, these 3 molecules of C4 compound is rearranged into the 2 molecules of C6 compound, fructose 6 phosphate. So by the action of this carbon rearrangement module, they can convert the 1 molecule of C6 compound into 3 molecules of C2 compound without any CO2 emission. But anyway, so yeah, it's a bit complicated to understand the whole structure of this pathway. But anyway, what is important is by using this metabolic pathway, just by using a very cheap substrate, I mean starch and inorganic phosphate, then we can produce ATP from ADP. And so we introduced 12 enzymes involved in this NOG pathway. All of them are obtained from thermophilic microorganisms, of course. And we introduced and we express all of these 12 enzymes in a single E. coli strain. And by doing this, just by cultivating 1 E. coli strain and harvest the cell and just incubate the crude extract of the E. coli at a high temperature, so we can very quickly prepare the cocktail of these 12 enzymes, which are consisting of this non-oxidative glycolytic pathway. So by using this heat purified enzyme cocktail, we checked the performance of our reconstituted NOG by using this reaction as a model reaction. So as you can see, that we could produce about 35 millimolar of phosphorylated glycerol using NOG, and actually the amount of ADP we put in the reaction mixture was only 0.1 millimolar. I mean, only using 0.1 millimolar of ADP, we could produce more than 30 millimolar of glycerol 3-phosphate, which means this reaction, phosphorylation of ADP to ATP, is cycled, carried out more than 300 times. So you can see this is a pretty nice turnover number of NOG to convert ADP into ATP. So this is a very good ATP regeneration system for our in vitro metabolic pathway using thermophilic enzyme. And actually, we also extended this NOG cascade for the production of some other useful compound. In this case, we applied we integrated this NOG cascade into this UDP glucose. This is a glycosyl donor used for a variety of glycosylation reaction. So this UDP glucose is used as a donor of glucose moiety to glycosylate the substrate of this enzyme, and they produce the glycosylated product like this. But after donating the glucose moiety, of course, this compound lost the glycosyl moiety. Then they have to be regenerated to provide another molecules of another glucose moiety into the substrate. So by integrating, I will skip the very detail, but by integrating this NOG cascade to this UDP glucose recycling module, we could achieve we could reconstitute this UDP glucose regeneration moiety. And actually, this is still under ongoing project. So the conversion yield is not perfect. It's still around 50% or something. But anyway, so we could demonstrate this UDP glucose regeneration pathway can work as by showing this model reaction, the glycosylation of glucose to produce this disaccharide trehalose. Okay, so the next is the last topics of my today's talk, NAD stabilization. So as well as ATP, the regeneration of NAD and NADH is a very important challenge to continuous implementation of in vitro reconstituted pathway. But in case of NAD, not only the balance between NAD and NADH, we found there is another very critical issue. That is the thermal instability of NAD. Because we are using the thermophilic enzymes for the reconstitution of our metabolic pathway, the reaction should be performed at a relatively high temperature, like 70, 60, or 70 degrees, because the thermophilic enzyme shows their highest activity around under high temperature. Nevertheless, NAD, the thermal stability of NAD or NADH, of course, NADP and NADPH, their thermal stability is not very high. So you can see here, if we incubate NAD at 60 degrees, they are gradually decreased, and their decomposition product, nicotinamide and ATP ribose, are accumulated in this way. So once the regeneration conversion between the NAD and the NADH or NADP and NADPH can be easily done by using some oxidation or reduction reaction. But if NAD is decomposed into totally different compound, it is very challenging to recycle this compound into NAD. And actually, the NAD and the NADP and their reduced form are involved in more than 20% of the metabolic reaction in living cell. So it is quite challenging to design in vitro metabolic pathway which is independent, which does not depending on NAD or NADP. So we have to manage this thermal instability of NAD. And actually, what we did to overcome this challenge is we again reconstituted metabolic pathway, in vitro metabolic pathway, for the salvage synthesis of NAD from its thermal decomposition product, I mean nicotinamide and ATP ribose. And actually, so this pathway is conserved not only in thermophilic microorganisms, but also in many living organisms, including us, human beings. So this kind of, the resynthesis of NAD from their decomposition product is most likely very important. They play a very important role, not only in thermophilic microorganisms, but also in the mesophilic organisms, including human beings. But anyway, so we isolated the enzymes involved in this and the salvage synthesis of NAD from thermophilic microorganisms and reconstituted this pathway in vitro. And if we compare the the concentration of NAD in the presence and the absence of this in vitro metabolic pathway, the result is very clear. If we have this in vitro pathway, the NAD concentration could be maintained at almost a similar level. Also, they are decomposed without the in vitro pathway. So by doing this, we could achieve this. Actually, this is not stabilization. The NAD is decomposed, but synthesized again. So we can say this is a kind of the apparent stabilization of NAD. But anyway, so by making this kind of in vitro metabolic pathway, we could achieve the apparent stabilization of NAD at high temperature. So, and as I said, so this NAD salvage pathway are conserved in a variety of living organisms, not only in thermophilic microorganisms but also human beings or other the organisms living at the normal temperature environment. But nevertheless, we can easily imagine that the thermophiles, this kind of NAD salvage synthetic pathway plays much more important role in thermophilic microorganisms because they grow at a very high temperature environment, and the NAD is not stable at a high temperature. So to investigate, to assess the importance of this NAD salvage pathway in thermophiles, we did some knockout experiment using these thermophilic microorganisms, Thermophilus, which has an optimal growth temperature around 70 degrees. So what we did is we knock out the enzymes catalyzing the initial reaction of this NAD salvage synthesis, and we assess its impact on the growth temperature of Thermophilus. So first, we investigated the growth profile of the wild type and gene disruptant of Thermophilus at around its optimum growth temperature, 70 degrees C. So you may see that there is no significant difference in the growth profile of the wild type and the gene disruptant of Thermophilus. So actually, this result was very disappointing for us because we expected that there should be a significant difference, significant growth deficit in this gene disruptant mutant. But actually, we could not find any significant difference. However, when we increase growth temperature of these Thermophilus strains to 80 degrees, in the wild type, of course, growth rate is somewhat slower, but they still show significant growth. But in the gene disruptant, in which nicotine amidase enzyme is knocked out, they showed very slow growth rate, almost no growth. And if we put this compound nicotinate is actually the product of the enzyme which we knocked out in this study. If we put this nicotinate in the culture medium, their growth deficiency was somewhat recovered, which clearly means this growth deficiency comes from is a result of the knocking out of nicotine amidase genes. So these results clearly demonstrate the homeostasis, the maintenance of intracellular concentration of NAD is the very important factor for thermophilic microorganisms to survive at extremely high temperature. So in other words, we can say this is one of the mechanisms of thermophiles to adapt to a high temperature environment. So actually, this is not the this study was performed not only for the production of useful chemicals or this kind of application of biotechnology, but one of the good actually, so this is the kind of the fundamental study of microbiology. And this is my last slide in today's talk. So you may know who is him. Louis Pasteur, that he is a very famous microbiologist, the French microbiologist. So what he said is, so you can see here, there does not exist a category of the science which one can give the name of applied science. I mean, he said there is no word like applied science. So what we have is science, and also the application of science. And both the science and the application is something like the bond together as a fruit, and the tree which bear that fruit. So which means not only the application, but also the fundamental study is also important. So perhaps some of you may be very interested in the fundamental study or applied application of the fundamental science. But actually, to make some, good research, both application and also the fundamental science is important. So I'm happy if you put this word of risk pass-through in your mind. Okay, so now yes, so this was the last slide. So now I like to close my today's lecture. Thank you very much for attention.
Thank you very much, Professor Honda, for such an informative and inspiring presentation. Now let's move on the Q&A session for all participants in this room or in the Zoom platform. If you would like to ask a question, please raise hand, or you can write the question in the chat box. Don't forget to introduce yourself before asking your question. Okay, please, Alwa.
Hello, Professor Honda.
Yes.
My name is Alwa Faniam Putri. I want to ask something before that. Thank you for the outstanding presentation. There's something that raised my curiosity. The one question is the cascade pathway analysis for the enzyme expressed in E. coli has been conducted in vitro. What other house, such as Pichia pastoris, utilize the same in vitro construction? If not, are there any specific consideration that need to be addressed? And the second question, Rhodococcus opacus is used in water-free organic medium reaction, where enzyme in the organic solvent form small aggregates. Do the enzyme not undergo denaturation, or is there any specific treatment applied to the enzyme before use? Thank you.
Okay, so let me answer from the second question. And actually, so in this study, we did not introduce any special modification to the enzyme. We just recombinantly produced these two thermophilic enzyme in Rhodococcus opacus cell, and we put the wet cells of Rhodococcus opacus cells, recombinant Rhodococcus opacus cell in the reaction mixture. And yeah, actually, organic solvent is toxic, has some negative effect on the enzyme stability. But the thermophilic enzyme has a very high stability at high temperature, and not only against temperature, they also in general they also has a better stability tolerance against other the protein denaturation factors like organic solvent or detergent. So that is why of course, this reaction do not have to be performed at a high temperature. Even the mesophilic enzyme may also work, because actually inside of Rhodococcus is aqueous water environment. Nevertheless, some small amount of organic solvent may have to penetrate it into the cell. So we employed the thermophilic enzyme because they have, in general, better stability even against organic solvent. That is why we use the thermophilic enzyme. But actually, we did not make any modification in the enzyme. And for the first question, sorry, I could not fully understand your point. So you are going to use Pichia pastoris for the enzyme production?
Yes, Honda. If we use Pichia pastoris as host, in the in vitro construction have a different with the in-host E. coli.
Yes, maybe it will also be possible, but maybe you have to consider what is the strong point of your host strain. The E. coli is very, in case of E. coli, we have a variety of genetic engineering tools. So, for example, even if the recombinant thermophilic protein cannot be correctly folded in E. coli, we may be able to introduce the chaperone protein or other the protein folding tag into E. coli. This kind of genetic engineering tools are very abundant in E. coli. This is the strong point of E. coli. And in case of Pichia pastoris, their strong point may be, for example, their very good ability to produce extracellular enzymes, secrete out the recombinant protein, or they are also able to mediate the protein glycosylation. And because thermophilic enzymes are mostly from bacteria or archaea, they do not require glycosylation modification. In this term, from this aspect, we can say the use of Pichia pastoris may not have the very big advantage. But if we want to use the extracellular protein, the Pichia pastoris, or for example, Bacillus, it is a bacterium. The Bacillus also has a good ability to secrete the protein. So if we want to use the thermophilic enzyme, which is produced outside of the cell, employment of Pichia or Bacillus, this kind of microorganisms may have some benefit.
Please for Ibu Catur Riani. Is there any question, Bu? Ibu Chaturiani, please.
Maybe or she has some problem in audio. I cannot hear.
While waiting for the question from Ibu Chaturyani,
Ah.
is a question from Alfi Rahmi.
Okay. In chat.
Yeah, thank you for the great presentation, Sensei. I'm Ami. I would like to ask, are there specific challenge in maintaining enzyme activity after immobilization? Thank you.
Okay, thank you very much. It's a very good question, and yeah, enzyme immobilization is very important challenge to make it possible the enzymes to be recycled, because we are using thermophilic enzymes. They have the very good stability. So enzyme immobilization and their recycle is very important. Of course, there are so many different ways to immobilize the cell, immobilize the enzyme. So what we did so far, one of our challenge for enzyme immobilization is like this. So, in this study, we tried to use the whole cell of E. coli after the heat treatment. So by incubating at a high temperature, E. coli is of course killed, and most of the enzyme inside of E. coli are denatured. And we can use those heat-treated E. coli cells as the kind of immobilized enzyme. But the problem is, so if we heated incubated E. coli cell at very high temperature, so maybe you can see here, the membrane structure of E. coli is destroyed, and enzymes are leaked out from the cell. So before the heat treatment, we put some small concentration of the chemical reagent, glutaraldehyde, which is often for the cross-linking of protein or other biomolecules. So after the glutaraldehyde treatment, E. coli could maintain the healthy structure even after the heat treatment. And of course, the E. coli are already killed at high temperature. Nevertheless, the enzyme inside of the thermophilic enzyme inside of the cell could be remained. And by using this, the GA glutaraldehyde treated and heat treated E. coli cell, we could perform some enzymatic conversion. So this is one example, but actually, so other immobilization technique can also be applied. Yes, so this is the answer to question one. And now Ryan-san also, Kachua-san, kind of chaperone normally for correct folding some. Okay, she is asking about the protein chaperone. And yes, yeah, this is also a very important question. And sometimes we can use very conventional chaperone protein like GLO-EL. And there is some commercial product of the chaperone protein. And yeah, of course, sometimes we can use them for the correct folding of some protein in E. coli. But generally, the thermophilic microorganisms also produce some chaperone protein, and the co-expression of those thermophilics, the chaperone proteins sourced from thermophilic microorganisms is, yeah, actually it's case by case, but in my sense, they have a better performance than the normal mesophilic chaperone protein like GLO-E or GLO-EL. So yeah, actually, the use of the chaperone protein from the thermophilic microorganisms is better. as my impression. Maybe there is no general answer.
Okay, next, there is a question from in this room. Please, Gita.
Okay. My name is Nita. I want to ask about NAD homeostasis. You said that NAD homeostasis is one of key factor for thermophil's organism to survive at extremely high temperature. Does it apply to all thermophilic organism? And how about the acidophilic organism? As we know that many thermophilic organism is also acidophilic too. Thank you.
Okay, thank you very much for the question. Yeah. Very honestly, I don't have any answer because what we investigated is only this thermophilic bacterium, Thermophilus. But if we look at the genomes of thermophilic microorganisms, most of thermophiles are equipped with this NAD salvage pathway. But the point is not this pathway is conserved not only in thermophiles but also the many mesophilic microorganisms or higher organisms like human beings are also equipped with this NAD salvage pathway. So probably it may be possible that not only thermophilic microorganisms, but also some mesophilic in some mesophilic microorganisms, this NAD salvage pathway may be a key factor to decide the upper limit of their growth temperature. And for acidophilic microorganisms, this is also a very interesting question. Today, I talked about only the composition of NAD into nicotinamide and ADP ribose. But if we change the pH under a very acidic condition or very alkaline condition, NAD or NADP are decomposed into different compounds. So if they are decomposed into different compounds, of course, this pathway cannot work. So yeah, I have no answer, but I suppose their acidophilic microorganisms may have the different pathway to desynthesize from NAD or NADP from different decomposed product. This is a very important challenge.
Okay, is there any question for all participants in the Zoom platform? All right, Prof. I believe the Q&A session time is enough. The time is now so 10 p.m. We have reached the end of today's guest lecture. I would like to extend our deepest gratitude to Professor Honda for sharing the knowledge with us. And thank you for everyone for joining in and asking great questions. I hope today to give you some new ideas and inspiration. And now I will return to our MC, Salwas.
Thank you to Devianti Nur Afifa for skillfully moderating today's discussion, and to Professor Kohsuke Honda for delivering such an insightful and valuable lecture. Ladies and gentlemen, we have now reached the end of today's class. We hope the knowledge shared today will be beneficial and contribute to our understanding. For closing, we would like to present a certificate of appreciation to Professor Kohsuke Honda for his participation and for sharing his expertise in this lecture. Please give applause. Once again, we sincerely thank you for the knowledge and insight provided. We also extend our gratitude to all participants, both those who join us offline and online platform, for your attendance and active engagement. On behalf of all the organizing committee, I, Salwa Fania Putri, would like to apologize for any shortcoming during the event. We look forward to seeing you at our next lecture series. Have a great day and enjoy the rest of your activities. Thank you very much and good morning.
Thank you very much.
Thank you very much, Prof. Thank you very much.
Thank you very much, Professor.